Welcome to the SPB SFX instrument, the home of structural biology and more at the European XFEL. What we do here is we look at the structure of predominantly biological samples towards atomic resolution to understand what they look like and hence how they work and function. One of the reasons you might want to use an XFEL to look at these things is to look at structures that might be radiation damage sensitive and not easy to look at in a synchrotron, or to look at, say, non-crystalline samples where we don't have enough photons at other sources to see them. Here at the SPB SFX instrument, we focus the FEL beam from the European XFEL down to a few microns or a few hundred nanometers, depending on the experiment. We scatter those X-rays off the sample in the forward direction and catch them in our AGIPT, our fast megahertz repetition rate detector, downstream to measure diffraction data, which we can then invert and understand as the structure of the sample in question. There are two classes of experiments that we predominantly perform here at SPB SFX. The first is serial crystallography, where we take a series of small crystals or radiation damage sensitive crystals, or perhaps even optically excited crystals, and look at those sequentially with the XFEL beam as they fall into the beam in a liquid jet, one after the other. And by collecting many patterns of diffraction from those crystals, we can infer the structure of the system under study. And of course, we can also infer the time-resolved structure of the system under study if we excite those crystals, say, with an optical laser, or if we mix in a substrate or a drug and watch how these things unfold in time. And it's the high repetition rate here at the European XFEL which gives us a competitive advantage compared to all of the other XFELs on the planet operating at the moment. We also do single particle imaging experiments where the idea is to shoot in small non-crystalline biological samples and actually increasingly also inorganic samples. We scatter X-rays from those, measure them also in the detector, and infer the structure from that as well. And here we've performed experiments in both of those modalities and more to understand the structure of matter at those length and time scales that are potentially inaccessible anywhere else. The tank you see in front of you is the 100 nanometer scale mirror chamber of the SPB SFX instrument. It's essentially a KB mirror system with a focal length of the order of 2.5 meters, a bit more for one mirror, a bit less for the other mirror, and it produces a spot, maybe a few hundred nanometers in diameter in the upstream interaction region. And these mirrors are predominantly used for experiments that require high peak intensities like single particle imaging, and very conveniently. Upstream, in another hutch about 30 meters upstream, is a mirror system, a 4-bounce mirror system that produces a focus that's about 10 times larger, about 3 microns in diameter in the upstream interaction region. We use that focusing system predominantly for serial crystallography experiments. The mirrors are probably amongst the flattest objects on the planet, with a deviation from perfect, not much. Maybe enough that we can see it, we're going to measure it, but not much at all. We're looking at the upstream interaction region of the SPB SFX instrument. And this is where the magic happens. So the sample is delivered from above for a serial crystallography experiment. That's typically in a liquid jet. It's injected into this catcher that you see here, and the sample goes through and is caught actually in the bottom. The X-rays come from right to left. And they pass through this optical microscope and they're focused inside the catcher. And there is the interaction region or the interaction point where the X-rays meet the sample. Then on the occasion that there is a crystal within the beam, that crystal diffracts and is measured in a detector downstream. The same basic principle also applies for looking at non-crystalline samples, except we typically inject them with an aerosol. That aerosol also is squirted into the catcher. The focused X-rays interact with ideally a single particle of the sample under study, perhaps a virus, and that is scattered into the detector as well downstream. All of this happens at a megahertz repetition rate, or perhaps even up to 4.5 megahertz repetition rate in the case of single particles. We're yet to try all the way, but both methods have already been demonstrated at 1.1 megahertz repetition rate. We have other apparatus, including inline microscopes, side microscopes, coupling for optical lasers that support those activities as well. This large piece of apparatus that you see in front of you is our megahertz 1 megapixel detector. It's called AGIPT, the Adaptive Gain Integrating Pixel Detector, and it's capable of measuring at up to 4.5 megahertz repetition rate. This detector can measure up to 352 pulses per train, and we have 10 trains per second. It's giving an average repetition rate of up to 3,500 frames per second. So I like to call it the fastest X-ray detector, direct X-ray detector in the West, or anywhere actually. What's nice about this detector is it's sensitive to single photons with a pretty good fidelity, and it also has a high dynamic range. The former is very important for single particle imaging experiments where we have very weak signal landing on the detector and we want to discern each and every photon and make sense of it. The latter is very important when we have bright signal from the Bragg peaks produced by serial crystallography experiments, and it's incredibly convenient that we can use the same detector for both experiments. This detector has been involved in, at time of recording, almost every paper produced from the instrument to date, measuring either crystallography data or single particle imaging data. What you can see here is the pulse arrival monitor. This is a device developed with the diagnostics group along with the SPB SFX group that measures the difference in arrival time between an optical laser and the X-ray pulses. We can do all the different kinds of things that you would imagine one wants to do with pulse arrivals. What's nice about this particular diagnostic is it can be in the beam while we're performing an experiment, so it's a true diagnostic. The other thing about it is it's compatible with the megahertz repetition rate, and so we can use the train and we can use the beam for a science experiment while being able to measure the delay between optical excitation and X-ray probe. And if you want to know more, have a look at our paper in Optica, published in 2020. So downstream here we see the refocusing compound refractive lenses, which refocuses the beam so we can use it again for another experiment downstream. This apparatus was contributed by the SFX User Consortium project. We have refocusing lenses, we see a second interaction region, and we have another detector, this time the Jungfrau Detector, to measure scattering as well. In principle, we could perform then 2 experiments in parallel, or more conveniently, what we can do is perform very different experiments in a different setup downstream.